Today's foray into our microbiology text by Tortora takes us to chapter 18, where we'll be looking at some applications of immunology that microbiologists can use for diagnosing disease. There's just our standard opening picture showing some Bordetella pertussis bacteria. Just for a little history, smallpox was a terrible disease that we've mentioned in the past that had a super high mortality rate, and it was a worldwide problem, and so it was a source of, or a subject of, a lot of research and interest in prevention. And many centuries ago, even in parts of Asia, they had discovered that inoculating someone with some material that came from scabs from sores produced by this bacteria could prevent the disease. Nobody knew why. They would just take some knives and cut a bunch of small cuts in the skin so they're all bloody, and then put some of this material in there and let it sit there and incubate and then heal up, and people would become immune. Well, this was sort of specialized into what was called variolation, in which they had a little bit more of a standardized method by the 18th century. And then finally, vaccination was introduced when Jenner observed that women who milked cows for a living or for part of their job on the estate were immune to smallpox. And there was a similarity between the cowpox virus, a relatively mild disease-causing virus, and the smallpox virus. And he developed a vaccine so that, and he actually surprisingly tested it without 100% knowing what would happen by inoculating a child with cowpox and then exposing him intentionally to smallpox. And luckily, the child survived, was protected. That brought on a revolution of the idea of vaccination. Louis Pasteur discovered a way of inoculating or vaccinating people after their exposure to rabies. Rabies is a slow-growing and slow-progressing disease initially. You can actually vaccinate someone after they've been exposed to rabies and have success with producing immunity during that incubation period. So he found a way to use a virus that had been reduced in virulence. That's a common thing to do in producing vaccines. Take a virus and grow it in such a way that it loses a lot of its disease-causing power, but yet it still carries a lot of antigens, and then infects someone with that virus, and then that will produce excellent immunity, but not cause severe disease. And his rabies was such a terrible disease and such a big problem worldwide that rapidly, after demonstrating the success of his rabbit nervous system rabies vaccine, all over the world, clinics popped up in major cities to recapitulate the successful treatment. All right. Well, how important are vaccines? Here's just a quick example. There's a lot of controversy these days about our vaccines, and I think it should be quelled by looking at some data like we see right here, in which we see the incidence of reported number of cases of measles in the United States in the 1960s, and then the vaccine was introduced, and you can see here by 1995, by 1990, there's essentially there's a eradication almost of the disease. This inset shows that we're not talking about the true eradication. There are still a handful of cases every year, and periodically people get the notion that they know better, and that that vaccinations are not only not important, but they're dangerous, and they refuse to become vaccinated or have their children vaccinated. And what happens is we have growing population of susceptible people, and so we have little spikes in the vaccine. Remember, measles can cause a fatal encephalitis, sclerosing panencephalitis, which is a terrible, terrible thing to have happen. And so there's a reason for trying to reduce the incidence of this disease in the population. Tremendously effective strategy. That's just one example among many of diseases that have been essentially eliminated in this country by vaccine production. And here's a here's a world map showing the countries with the highest measles mortality rate where there still isn't um, broadly used vaccination. So you can see we have a long way to go. We need to get these you know, medical utilities out into the world more in order to help improve the quality of life for people around the world. Pretty severe disease still. No matter where I go, I'm going to be on top of some text, aren't I? Um, Here are just some examples of recommended vaccines by the CDC. All these vaccines on this table and on the next page, most of the ones on the next page, are all vaccines that are recommended for infancy, childhood, and teen year vaccinations. And so these are some really important diseases that you probably don't know anything about because people are vaccinated for them, and we just don't have these diseases in our population. Beforehand, before the vaccine programs were introduced, these were all severe diseases that caused significant mortality rate among children and young adults. Diphtheria, it's a toxoid vaccine, meaning the diphtheria toxin, which kills epithelial cells, is denatured and then used to produce a vaccine. Meningitis is a life-threatening, very fast-progressing bacterial disease in which the bacteria get into the meninges, the coverings of the brain and spinal cord, into the cerebrospinal fluid and so forth, and cause death within a few days. And so rather than continuing to there was a time when they didn't have a vaccine for this, and there was just a lot of effort made to educate young college students coming into college and their families about meningitis so that if anyone developed symptoms, they would know really quickly to be tested and treated. But now we have a very good quality vaccine against the Neisseria species that cause meningitis, Neisseria meningitidis. It's a conjugated vaccine, which is interesting. It's made from polysaccharide from the capsule of these bacteria, but all by itself, that polysaccharide isn't very immunogenic. It doesn't produce a very vigorous immune response. And so they take the diphtheria toxoid, same thing that's used for making the diphtheria vaccine, and then covalently attach that onto some of the polysaccharide from the meningococcal cells, and that produces something that's very, very immunogenic. And while the immune system's all revved up, you're attacking that diphtheria toxoid portion. Excellent antibodies are made against the polysaccharide components. So that's been a great discovery in how to make the capsules polysaccharides useful for vaccines. Pertussis is the vaccine that's typically used in what's called the DTaP vaccine, diphtheria, tetanus, and acellular pertussis. It's just fragments of Bordetella pertussis bacteria containing the proteins, the antigens. So it's not truly a toxoid, but it contains toxoid, and it contains all kinds of bacterial antigens. Pneumococcal pneumonia, Streptococcus pneumoniae is the most common agent that causes pneumonia. And as in many cases of bacterial, various types of diseases, the most susceptible populations are the very young, the very old. So we have a vaccine, a conjugated vaccine, same exact conjugating protein, the diphtheroid toxin, used to make this capsule vaccine, anti-polysaccharide vaccine. Tetanus, you know all about the tetanus. Clostridium tetani produces that terribly powerful toxin, while the toxoid vaccine is effective against tetanus. Haemophilus influenza type B also produces meningitis. So there's a conjugated vaccine against the Haemophilus influenza capsule as well. Influenza is an inactivated virus. So virus, as we'll see in a subsequent slide, is grown up in the factory and then heat killed and then used as a vaccine. Every year, a new influenza vaccine is produced against the most prevalent 3 strains of flu virus around the world around the time of the beginning of the flu season. And then that's given to as many people as will be willing to take it for free. And it's very, very helpful to reduce the incidence of influenza. Influenza is a pretty serious disease. I think oftentimes people say, oh, I had the flu. I think what they really mean is they had a bad cold. Influenza is pretty rough stuff. relatively speaking, and a very uncomfortable and unpleasant experience. Measles, mumps, and rubella MMR vaccine. It's attenuated viruses. These are viruses that have been grown in culture for a long, long time to the point where they just don't produce any strong disease anymore. But nice thing about them is they produce the most long-lasting immunity of any type of vaccine. So you actually have live virus. Well, they're not really live, but you have active virus that can be injected but doesn't cause disease, but yet carries the antigens of the virulent forms. So that's the MMR vaccine. Mumps is another one, and rubella produces German measles. Chickenpox, the varicella vaccine, human herpes virus 3. There's a vaccine against chickenpox so that people don't have latent HH3 viruses living in their dorsal root ganglia of their sensory neurons, and they don't get shingles later in life. So they eliminate the chickenpox experience and also prevent the possibility of shingles. Poliomyelitis, terrible disease that was a big, big problem, and I wish I had my wife here to tell you the story of how the vaccine was successfully prepared, developed, and to root out that disease from our society. Very interesting story indeed. If you ever want to Google that, there's a movie actually available about it that's worth taking a look at from a different era. Killed virus. All right, rabies. The rabies vaccine is not among those. All the ones we've been looking at are ones that are recommended by the CDC for everyone to have during infancy, childhood, and adolescence. A rabies vaccine is used after exposure to the virus. Again, as I mentioned, you can successfully vaccinate after exposure in the case of ELISA virus. Hepatitis A and B are recommended. Hepatitis B, especially for anyone who works in a healthcare setting, you must by law be offered the hepatitis B vaccine. Hepatitis A vaccine, a very good idea. Smallpox, how interesting is this? Smallpox is the only major disease that's been completely eradicated from the world's population. There are no areas in the world where smallpox is endemic or even sporadic. It's gone, and so you don't even immunize anyone for smallpox anymore. So it almost seems scary, but they're even just as long ago as now don't say anything smart, Alec. As long ago as when I was in childhood, everyone got a smallpox vaccine, and that's no longer the case. Probably none of you had a smallpox vaccine. Herpes zoster, it's the same virus that causes chickenpox, but when they say herpes zoster vaccine, what they mean is a vaccine that's intended for adults who have had chickenpox so that they don't get shingles later in life. So that's a big problem. Now that we have an aging population, shingles is a very miserable disease. And part of the problem is that even after the couple of weeks of very, very painful lesions that are produced by the reactivation of the virus, maybe as many as years or even decades later, pain is still experienced. So this is a big problem with this disease. So a vaccine has been produced. Human papillomavirus is recommended. It's papillomaviruses cause benign things like wars, but they cause cervical cancer as well. That's a pretty significant frequency of disease that's been pretty much. Hopefully, we're going to be able to knock that back. So here's a list of recommended diseases or vaccines again for children. You can see that same list we've just been looking at, and what kind of vaccines they are. And a few more of the ones that were on the list are continued to be given as multiple doses throughout, all the way through the age 18. All right. So that's what public health is all about. The government, which some people are very afraid is infringing on their rights, you can see what side I come down on, doing things that are proactive to improve the health of the population. I think that using the science that we have, excellent science that we have today, in the pursuit of better living for the population is well worth it. You saw what happened to the incidence of measles, and just every single one of those diseases on the list of vaccines we just looked at is the same story. Pretty much reduced the incidence in the country to essentially nothing by routine vaccinations. So types of vaccines. Attenuated whole agent vaccines are the best kind of vaccines. They produce the longest lasting and most vigorous immunity. So again, growing bacteria or viruses in culture for a long, long time where they're not needing to have all their virulence factors to be able to survive in the body, they tend to drop them off. And so they become less and less virulent, less disease causing, and yet they still have antigens that will produce excellent immunity, but they actually infect the body and produce the best active immune response. Polio vaccine, there's multiple polio vaccines. There's an inactivated whole agent vaccine, and then there's another vaccine that's not with whole agents, not live. That's also very useful. Toxoids, again, just the toxins produced by this bacteria that are denatured so that they're inactivated, either treated with heat or with formaldehyde or something to ruin them, basically, so they can't bind to their cellular receptors and cause disease, but they still cause excellent immunity, produce excellent antibody responses and immunity. Subunit vaccines, that means something from the agent has been purified out, maybe even in a fairly coarse way. Take a whole kettle full of the microorganism and then kill it, bust it up, and then just use some biochemical strategy to just capture some fraction of that material that's good for producing immunization, and that's called a subunit vaccine. It could even be a purified protein, but it could be something a little less neat and tidy as all that. Acellular pertussis isn't a single individual protein, but it's some components of the destroyed bacteria that are used for the vaccine. Nucleic acid vaccines, I don't think I have to bring that up for you, but for the very first time in the context of our recent or ongoing COVID pandemic, RNA vaccines were used for the first time. So recombinant, this example is talking about recombinant DNA vaccines, but actually recombinant RNA vaccines turned out to be very effective and relatively affordable to make and very quick to make and to modify for a virus that's changing by mutation very quickly. Here's somebody embryonating an egg. So you can inoculate or inject antigens into, or viruses, I should say, live viruses into an egg embryo and produce large quantities of, in this case, influenza virus, and then you can purify that out and use that to produce vaccines or tests. New vaccines. Culturing pathogens is the strategy typically. We talked about RNA and DNA vaccines, recombinant vaccines. Adjuvants, what's an adjuvant? Oftentimes, even if you make an excellent antigen preparation for vaccination, you get a much better response by the adaptive immune system if you add just a random other protein that's already known to get the immune response really revved up. Some proteins, for some reason, just really get the adaptive immune response and inflammation ramped right up. If you mix that together with your vaccine, you get a better outcome. So adjuvants are just chemicals and proteins and lipids that are used together to help boost up the immune response to a particular vaccine. Therapeutic index. Any medical treatment, no matter what it is, has some element of risk associated with it. There's always a downside or a side effect that needs to be taken into consideration in terms of assessing whether it's a reasonable choice for a particular malady. And it's no different with vaccines. There's a small percentage of occasions in which people respond allergically to vaccines. Other side effects that may occur with various vaccines. With live attenuated vaccines, it is possible for a vaccine to revert to a more virulent form. And so on rare occasions that can happen. So always you have to look at the severity of the disease and determine, do the risk-benefit analysis and determine whether or not it's a good choice for the population or for anyone to make for protecting themselves against a particular disease. So next part of our chapter, we're going to talk a little bit about, just briefly, about introducing some different technologies that are used to apply what we know about immunology, the tools of immunology, to diagnosis of disease. And when we're thinking about diagnostic tests, we have to think about sensitivity. What's the likelihood that the test will be able to identify or recognize the presence of a pathogen in the context of an actual diseased person? If the test can only identify that level of concentration of a pathogen 10% of the time, it's not going to be very useful. We need a test that's going to be sensitive enough to always detect the presence of the pathogen in the context of typical disease. Specificity. We need to have a test that will identify only the pathogen of interest and not be fooled or affected by various other conditions or antigens that might be present at the same time, producing what are called false positives. We don't want to have a test that's going to light up and say, yes, the pathogen is present when it really isn't, because then we'll subject people to treatments that may be expensive or have their own side effects and risks associated with them. So we need good specificity. Immunologic-based tests are typically quite sensitive and specific, so that's one of the beauties of antibodies, and antigen-antibody interactions are generally very specific. So, tuberculosis tests. We do testing for tuberculosis in this country whenever someone goes to get a job. They get a TB test in order to, and that's one of the things that was done to essentially almost eliminate tuberculosis from our population. But it's a little trickier for a number of reasons when it comes to tuberculosis, and I think we've mentioned at least one, and that is that the treatment period is long because of the slow growth rate of the organisms, and so oftentimes people will not comply with a long-term 6-month long or longer antibiotic treatment regimen. And so what happens is we produce people that carry the disease that don't believe they're carrying it, as well as antibiotic resistance. Oh, here's a cool thing. Now, a lot of treatments, not just for infectious diseases, it turns out, but for reducing inflammation in chronic inflammatory diseases, autoimmune diseases, for treating different types of cancers, monoclonal antibodies. What's a monoclonal antibody? An monoclonal antibody is a single immunoglobulin molecule that's produced in pure form by a cell that can be grown in culture, and then you just grow a large number of these cells and they produce one thing, that particular antibody. You can purify the antibody, and monoclonal antibodies are very specific and very pure, and therefore don't produce a lot of unwanted side effects when they're injected and so forth. Although people can react to monoclonal antibodies, they are, after all, foreign to the body. So how does this done? Inoculate a mouse with an antigen, right? It's a foreign substance, usually with an adjuvant, to try to really rev up the immune response, and then a whole bunch of lymphocytes will be produced that are making antibodies against that antigen, and a lot of them may be found in the spleen. The spleen's a nice big organ just packed with lymphocytes. It's lymphoid tissue largely, and so we'll take that spleen out, grind it up, and release out all the lymphocytes from the spleen, and put them in this test tube that we see here, and then take some cells that are called myeloma cells. It's a type of B-cell cancer. You can grow the myeloma cells in culture forever. It's called a cell line. Mix together the myeloma cells with the lymphocytes from the mouse, and you can induce the fusion of the myeloma cells with the lymphocytes. So just two cells will fuse together into one large cell, and then once you have that happen, you can grow those fused cells in tissue culture, and they'll produce antibody. The last step is you have to have a selection method so that the only cells that you grow in culture are the ones that have successfully fused myeloma cells with lymphocytes from the mouse, and then you have to screen all of those hybridomas, they're called, for the production of an antibody of interest. So there's that screening, the selection step and screening step come after this mixing together step. The selection is shown down here. The cells that didn't successfully fuse and produce a hybridoma are killed or don't grow by means of selection. We're going to screen. We take a whole bunch of, dilute all the hybridomas. I would suggest a better way to picture this is to dilute out the hybridomas so much so that in a 96 or 384 well plate, only one cell appears in each well, 0 or 1 cell, and then you can let those grow in culture, which is why you see multiple cells here, and then screen them for antibody. And boom, you have a source of antibody. Now you can grow it up in large cauldrons in the factory and produce enormous amounts of antibody to make into medicines or treatments. So that's become a really huge part of new medical modalities or treatments for a lot of different things. Here's just some examples. Miramonab CD3. It's a antibody against an antigen that's found on a particular subset of T cells that destroys transplanted organs. So it helps reduce the rejection of kidneys after a kidney transplant. Infliximab is an antibody against tumor necrosis factor alpha. And TNF alpha is a major inflammatory cytokine released by activated macrophages. And it turns out that that is one of the key activators of inflammation in chronic diseases like Crohn's disease, which is an ulcerative disease of the intestines in people with this. It's an autoimmune disease. And so this antibody binds up the tumor necrosis factor so it can't bind to its receptor on its target cells and cause inflammation. Remicade is called, I believe. Comalizumab is an anti-IgE antibody. If you remember me talking to you about last chapter about what happens when, was it last chapter? Anyway, what happens when people have an IgE response to an antigen, immunoglobulin type E's remain afterwards attached to mast cells, so the next exposure to the same antigen causes IgEs to trigger degranulation of mast cells, release of histamine, and massive inflammation. So very much unwanted allergic responses, and so using an antibody against IgE can block that response. There are some side effects that are still being, the medicines are being improved to help reduce the amount of the number of individuals that have side effects from macrophages taking up the comalizumab / IgE complexes and getting all wigged out. Rituximab is an antibody that's used to combat B-cell lymphomas. It's against an antigen called CD20 that's on the surface of a lot of B cells, but it's also retained on the surface of B-cell cancers. So long-term treatment with rituximab can reduce the presence of B-cell cancer cells in the body. Rheumatoid arthritis is a B-cell mediated autoimmune disease. So again, knocking down some of those some of the number of those CD20 positive B cells can help reduce the inflammation in that disease. This last one is an antibody against a growth factor receptor. So a growth factor that binds to a growth factor receptor that's important is certain breast cancers are Herceptin positive or sensitive, and this antibody can help get rid of those cells. As monoclonal antibody technology continues to improve, monoclonal antibodies or chimeric antibodies, they're called, are more and more becoming the mainstay, in which we have an actual human antibody with a different tip that with only the variable regions come from the mouse that was actually used to produce antibodies against a particular antigen. So you can have mice that have been kind of humanized so that the antibodies they produce are like human antibodies. There's a whole bunch of different strategies that range in the amount of human likeness of the antibodies, of monoclonal antibodies, to reduce the foreignness of the antibodies, to reduce the kinds of side effects that are associated with their infusion. You have to infuse these antibodies into the circulation of the patient on a periodic basis to get the results. Some other assays for antibodies and antigens. Here's an example of what's called a precipitation ring test. We have some soluble antigens and some antibodies. When you mix them together, so maybe you have some antibodies in this test tube, when you add a patient sample and you want to see if it has antigens, if the two meet each other, they'll produce what's called a zone of equivalence. And that's where a whole bunch of antibodies and antigens form a big lattice and they precipitate. It's exactly the same as the way we talked about antibodies precipitating antigens in the body to help rid the body of antigens in the case of disease. So this whitishness is precipitation that's happening in the zone of equivalence. Only a certain ratio of antibody to antigen will cause this precipitation to happen. So that's why we're in this tube. We're not going to mix vigorously. We're just going to let the antigens diffuse down into the solution. And at the right spot as they're diffusing down and we get the right ratio of antibody and antigen, we'll see that precipitation ring and know that there was in fact antigen in our test sample. So this is just showing you zone of equivalence occurs at just the right ratio of antibodies and antigen. We have that precipitation happening. If you have too much antibody excess, you won't have a precipitation reaction. If you have too much antigen excess, you won't have precipitation. It's got to be in that right ratio. Here's another test, a Western blot that's used for, in this case, the example uses for testing for Borrelia antibodies that against the bacteria that causes Lyme disease, but it's also a standard test for HIV infection. And so what you do is you take all the antigens from Borrelia and separate them by electrophoresis. So we have a bunch of Borrelia growing in a cauldron, bust them all up, separate their individual proteins. Like we put a little well at the top of this gel, would be all the antigens from Borrelia. And then you apply an electric field from top to bottom of this gel. The gel would be trapped between two glass plates. Excuse me. And then you just set up an electric field, and the proteins have electric charge, and they will move in the field as they travel down through this gel. They'll separate because every protein has slightly different properties. They move at different rates depending on their size and the amount of charge. So we have all these proteins separated into bands of purified protein, and then we transfer all that protein onto a piece of specialized paper, nitrocellulose or nylon paper, and then we add the patient's sample to see if it has any antibodies against one of those Borrelia antigens. And if there's anywhere there was an antibody from the patient's plasma that recognizes Borrelia, it'll stick to whichever protein antigen band is in the paper, the nitrocellulose, and then we add a second antibody which recognizes the human antibodies, which has an enzyme attached to it or a dye, and we can see right where it is and find out whether the person had any antibodies in their blood at all. Very, very sensitive test actually, and quite standardized, quite readily completed well in virtually any laboratory that can do testing. Agglutination reactions. If you have a bunch of, in this case, bacterial particles, if there's antibodies present, mix them together, and if you have the right ratio, you can have agglutination, clumping of the bacteria. So that's a agglutination reaction for antibodies. You can do an agglutination reaction for antigens as well. If someone has a particular antigen in their body because they have an infection, you can have latex beads coated with antibodies. So instead of having it this way, we have actual particles that are antigens, particles that are coated with, take some microscopic latex beads that are commercially available, coat them with antibodies, mix them with the patient's sample, and if they have the antigen present, it'll form agglutination. So that is, call it an indirect, positive indirect test. Here's a positive indirect test for antibodies. Again, we can coat the latex beads instead of using actual, in this case, actual bacteria. We can use latex beads coated with a particular antigen and then mix it with the patient's sample to see if antibodies are present, in which case they will cause agglutination. So we can test for either antibodies or antigens with these latex bead agglutination tests. Pretty cool. Here's a direct agglutination test. And so what we're going to do is put a series of dilutions of the patient's plasma into each one of these wells, and then we're going to add some red blood cells. In this case, it's a type of antigen that can cause red blood cell clumping or agglutination all by itself. And whoops, I'm kind of covering up some of the text down there. In this case, if there's the right amount, the right ratio of antigen to antibody, we'll have agglutination, a large mass of clumped antigen and antibody together, whereas if there's not enough antibody present, then all the red blood cells will just pile up at the bottom of the well. So here's a positive well in which the agglutination, it's all covering the whole bottom of the well is covered by this mass, this mat, as they say in the text, of agglutinated red blood cells. Whereas once we get out to, these are further dilutions, 1 to 20 dilution of the plasma, 1 to 40, 1 to 80, keep doubling every time we take a half a volume from this well and add it to this well, and half a volume from this well to this well, then adding the standard amount of blood cells. And at some point, the patient's sample is so dilute that it can no longer produce any agglutination, and that's what we see here. So that's a way of measuring the titer. What's essentially the effective concentration of antibodies that was present in the patient's sample? It's kind of arbitrary. It's not an actual concentration value, but in this case it was a 1 to 160-fold dilution that still produced agglutination, whereas a doubling of that dilution, 1 to 320, did not any longer produce any clumping. So if this is a standard test that everyone uses, doesn't matter that it doesn't have units of actual concentration. It gives the physician a sense of what is the titer of antibody in this person's blood. Hemagglutination. Some viruses like influenza will agglutinate red blood cells, and if you mix that with a patient's sample and it has antibodies in it, that will block the agglutination. So here's a little schematic diagram. So here's some red blood cells plus virus, and you mix them together, and it causes agglutination. So the CDC used to send out kits all over the world that contain these two things in it, and you mix it with the patient's sample. If the patient has been infected with a particular influenza strain, then it will block the agglutination. It's called a hemagglutination inhibition assay. I don't know if they use it so much anymore. RT-PCR has become so standardized, and it's so sensitive and so specific. I think that has become the gold standard for identification of influenza strains. Here's another interesting test. Here's a susceptible cell, which a toxin molecule will kill the cell. If you mix them together, you can see an immediate effect in the morphology of the cells that have been damaged by the toxin. If you mix that with a patient's sample, and the patient has antibodies against that toxin, it will then, the antibodies will tie up the toxin, and the cells will be protected, and you'll see a unchanged cell. And just another interesting application of immunology to diagnosis of disease. Here's one that's a little more abstract, not really bad or anything, but it's just kind of funny in a way. If you mix together an antigen and complement along with antibodies, and so now you have all the antigen and antibodies tied up together, right? And then you add some sheep red blood cells and antibodies against sheep red blood cells into the mix. Nothing happens to red blood cells because all the complement that you intentionally added at the beginning has been all saturated, all used up by the initial antibodies. So right, you choose the amount of complement you're going to add so that this works out well. So that in this scenario, there's no lysis of red blood cells by complement by the membrane attack complex. In a serum without an antibody, let's say there was someone who did not have the antibody in their serum, and you mix together antigen plus complement plus sheep red blood cells plus antibodies, the complement will lyse the red blood cells, and you'll have all the fluid, the solution phase will turn bright red from all the release of hemoglobin. So if the patient has antibodies in their plasma, you'll see no agglutination. If there's no antibodies present, you'll see lysis of the red blood cells. I said agglutination, I meant to say lysis. So that's kind of a cool, almost reverse kind of a strategy for testing for antibodies in someone in a patient sample. A couple of immunofluorescent methods. Fluorescence is a beautiful thing. You can see it very well. We talked about how fluorescence is used to increase the sensitivity and the minimum size, the resolution of microscopic methods. Well, here we have some group A streptococcus from a patient's throat. Mix that together with a fluorescent dye against a fluorescently dye labeled antibodies. I'll get it right. And mix them together. If the antibody recognizes the strep bacteria, the strep bacteria will look fluorescent. Now, this diagram makes it look like the antibodies are bigger than the strep, and you think, gee, how is that going to work? Actually, of course, these are single molecules that are on the surface of the strep, so they just coat the strep bacteria. So the bacteria just look the same as they normally look. They're just They just glow in the ultraviolet light, and the microscope makes that very beautiful. Of course, if there were no antibodies in the patient sample, then or no virus in the patient sample, then these antibodies wouldn't stick to anything. They would just remain like in background in the fluid, and you wouldn't see that bright color of the antibodies. Here's another one. You can look for syphilis causing bacterial antibodies. So if someone has syphilis, Treponema pallidum bacteria will have caused the induced an immune response, and they would have antibodies circulating. If you take some Treponema pallidum from the laboratory, grown up in the laboratory and culture, and then mix that with the patient sample, if there's antibodies present, then you can add a second antibody, an antibody against the antibodies, right? There's antibodies, you can make antibodies against antibodies. You inject some human antibodies into a rabbit, say, and now we got some rabbit anti-human antibodies, stick a fluorescent dye on there, and boom, you have something essentially what here only would have seen these spirochetes glowing with fluorescent light emission if someone was positive for syphilis antibodies. Flow cytometry, we talked about this once before. You can actually label cells with fluorescent dyes and then shoot them through a little nozzle to make little microscopic droplets that shoot in front of a fluorescent beam, or a laser beam, I should say, and of the appropriate wavelength to activate the colored fluorescent dye to emit light, and you can have a detector and count up the number of cells that are positive in terms of fluorescence and find out if someone has the requisite agent. So flow cytometry is a growing potential use in many, many laboratories. Flow cytometers that do this kind of thing are still relatively expensive, and it takes a relatively proficient technician to be able to properly run it. So they're not quite as readily used as some other methods we've been talking about. ELISA, we have looked at this before. Enzyme-linked immunosorbent assay is what that stands for. It's a kind of a sandwich assay, and you take a plastic plate with either square or round bottom wells like this, and you can either coat it with an antibody or with an antigen. In this case, we're going to coat it with an antibody first. It's called a direct ELISA to detect antigens. So we go into our freezer and get out an antibody that we have in the lab against a particular antigen, coat it. You can get it to stick to the plastic. Then you take the patient sample that may have some antigen. You're questioning whether it has antigen in it. And then wash, rinse away all the rest of the debris from the patient sample. Add to that a second antibody against the same antigen, only in this case it has an enzyme covalently attached to it. You can do that right in the lab. You can take a bunch of antibodies and attach an enzyme, a purified enzyme onto it. Then you take a substrate for that enzyme that turns color when the enzyme has reacted with it. And if there's color change, you know that the patient sample had that antigen in it. So I hope that's clear enough. We're just going to see if any of the patient's antigens stick to that first layer of coating of antibodies, add a second antibody, and get a color. We can modify this exact same assay. There's a million variations of ELISA, which makes it very, very effective for all kinds of applications. But in this case, we're going to go into our freezer and get out some antigen that we have all set aside and purified and make it stick to the plate. And then we're going to take the patient's plasma and add it. And if there are any antibodies against that antigen in the patient plasma, then it's going to stick to the antigen, rinse away everything else, and add a second antibody against antibody again in this case, and it has a dye linked to it, or an enzyme linked to it, I'm sorry, and then it'll cause that color change. So you can see that ELISA is very versatile. We can use it for these kinds of direct tests. You can also do ELISAs in which you are simply going to see if a competing antibody is present in the patient sample. You have added everything in the ELISA to make a color change, then you add the patient sample, and if they have antibody, it will compete with the dye-coated antibody, and the color will disappear, things like that. All kinds of different applications. Here we see an ELISA-like method that we're going to do right in a sponge, basically, and it's a pregnancy test, and there was an example of this already in your text. You may have looked at the PowerPoint before or already downloaded it before I modified it, but I think it was not at all clear, in my opinion, from the way they described it, how it was actually happening. So here's I think a better illustration. So here we have some antibodies in a foam, basically a foam strip, or a fibrous, porous, fibrous strip, and you're going to add a patient sample, right? That's what we're testing to see if someone's pregnant. Do they have antibodies against human chorionic gonadotropin? And we're going to stick that in there, or I guess antigen. And what happens is all the water that's in that sample starts to diffuse across to hydrate wet this dry slab of foam. And as the water migrates through this foam, if there's antigen, it sticks to these antibodies that are present already in the foam, and they are allowed to diffuse along with their antigen. And as they come along, there's some antibody in this strip right here that is fastened down. It's an antibody against the antigen, the HCG antigen also. And it turns out this first antibody I should have mentioned is got a color attached to it, a colored label of some kind, a blue label. And then so as the antibody antigen diffuses across through this foam pad, if there is in fact antigen present, it will get hung up here in this immobilized strip, and it'll accumulate and start looking concentrated, and that's when we'll see a blue line. The exact same type of test is used in the rapid COVID tests. You've probably done a number of those over the last couple of years. Only in this case, it's a COVID antigen that's being used. There's an antibody immobilized here, and usually those were a red dye attached to the antibodies, and it would show up in the test region. And there's a control strip there too. So that's a pretty fun application. Very, very elegant because it's very simple, easy to do, takes only a few minutes of time, and quite reliable for human chorionic gonadotropin. I have to admit, when I was talking about this one time, my student said, how come testicular cancer turns this test on? It's like, what? Somebody discovered, some man wanted to try his wife's pregnancy test and tested it, and it came out positive. And they said, now how can a man have a positive pregnancy test? And when they went to have it checked out, there were people that were very curious and were not willing to just sit back with this. And they found out that he had testicular cancer, and his cancer was producing an antigen that matched the antibodies in the test, and therefore came out positive. And so they may well have saved his life by him just randomly decided to do a pregnancy test on himself. It's a funny old world. So serological tests, typically when you talk about a direct test, you're testing for antigens in a patient sample, whichever one of these tests we've been talking about. An indirect test means you're testing for antibodies. But there's other uses of the term direct and indirect, especially when you're talking about competitive ELISAs, for example. So here's some of the tests we've talked about. Precipitation reactions, where antibodies and antigens, if they reach the zone of equivalent, will precipitate out and indicate that there's the presence of the agent that we were interested in. Remember the red blood cells in the serial diluted plate that we're agglutinating. Agglutination, particular antigens. We could take latex beads and coat them with either antibody or antigen and test for the opposite. Very effective and very versatile kind of a test. Hemagglutination, that's actually the one with the red blood cells that we're going to agglutinate with influenza virus. We talked about hemagglutination inhibition assays. So that was a big thing. I don't know how prevalent that is anymore. Neutralization, that one where the cells were going to be killed or damaged by the toxin, but not if there was antibodies in the patient's sample. It would protect the cells from the toxin. Fluorescent antibody methods. Those are very, very powerful. It's a standard way of detecting, to this day, standard way for detecting syphilis and rabies virus. There's a fluorescence test. Complement fixation. That was that funny one where if we mix together all the things that will cause complement activation and red blood cell lysis, unless there's antibodies present in the patient sample, in which that will prevent, it'll mop up all of the complement, and it won't be able to lyse the red blood cells. And then finally, ELISAs. Spacing out. What's that? I'm spacing out. I did ELISAs all through my graduate school years. Anyway, enzyme-linked immunosorbent assay. There we go. So here's some quick follow-up questions. See if you can answer these questions. Patient's serum, influenza virus, sheep red blood cells, and anti-sheep red blood cells are mixed together in a tube. Influenza virus does naturally agglutinate red blood cells. What happens if the patient has antibodies? That's that hemagglutinin inhibition assay. So remember that the control sample, if you mix together influenza virus with the red blood cells, then that will agglutinate automatically. But that's the control. If you add the patient sample, now it no longer agglutinates, and that shows the presence of influenza virus antibodies. Patient serum, chlamydia, guinea pig complement, sheep blood cells, and antibodies against sheep red blood cells mixed together in a tube. If there's chlamydia antibodies in there, that will prevent the lysis of the red blood cells because all the complement that was intentionally added as part of the mixture will be exhausted by all the antibodies in the patient sample, and there will not be any lysis of the sheep red blood cells. Whereas if there are no antibodies in the patient's serum, they always say serum here, that will cause lysis of the red blood cells. You probably saw I put a little complaint in the previous PowerPoint recording in which technically, physiologically, the term serum means taking a human plasma sample or an animal plasma sample, doesn't matter, the liquid portion of blood, allowing it to clot, centrifuging it down. All the fibrin, the blood clot, can be crushed down in a centrifuge to a little pellet at the bottom, and that fluid that's left is called serum. And in this context, immunologists tend to use the word serum to mean the liquid stuff that's taken out of someone's body as the blood, and then take the cells out. Not technically correct, drives me crazy, but that's the industry standard word they use. Serum, serum, serum. Never get used to it. Anyway, thanks for listening, and join me next time for chapter 19 about antibiotics.